Hi everyone, my name is Yann Deleye. I'm a postdoc in the White-Magara lab at Duke. I would like first to thank Phil to let me present at this year's Metabolic Physiology Isolation talk series. It's quite exciting for me because today I'm going to share with you a lot of unpublished data on the protein phosphatase M1K, PPm1K, a new regulator of protein translation and beta cell health. In the lab, we are studying how branched-chain amino acid metabolism is regulated, with really a strong focus on deciphering the link between dysregulation of branched-chain amino acid metabolism and metabolic and cardiovascular diseases. Myself, I am really interested in the role of PPm1K in type 2 diabetes. PPm1K is a phosphatase, dephosphorylating branched-chain keto acid dehydrogenase complex, BCKDH, and this event leads to increased branched-chain amino acid catabolism. So it has been quite intriguing and interesting to study the role of PPm1K in type 2 diabetes, because genome-wide association studies showed that the SNPs near PPm1K loci were the only one associated to all three branched-chain amino acids, isoleucine, leucine, and valine, and also that the SNPs near PPm1K were associated to increased risk for type 2 diabetes. Our group published previously that PPm1K does not only modulate branched-chain amino acid metabolism, but also PPm1K is a new phosphatase for ATP citrate lyase, and by doing so, restrict de novo lipogenesis in the liver. So based on these two roles from PPm1K, we first thought that PPm1K could be linked to type 2 diabetes due to maybe modulation of insulin resistance. However, genetic studies showed that these SNPs were not associated, in fact, to insulin resistance. The concept proposed is that there is a causal pathway from adiposity to insulin resistance to dysregulation of branched-chain amino acid metabolism. This leads to type 2 diabetes. Since the SNPs near PPm1K were shown to be associated to islets function, we believe that PPm1K not only in this context modulates branched-chain amino acid metabolism, but also that PPm1K in the pancreatic islet could play other role independent of BCAA metabolism, leading to type 2 diabetes. This study from Tanira et al. From 2012, in fact, supports our concept. Indeed, they performed an unbiased genetic correlation of type 2 diabetic genes, and they showed that PPm1K was associated with genes involved in insulin secretion in human islets. Moreover, they also showed that PPm1K expression was positively correlated to insulin secretion and negatively correlated to HbA1c. So altogether, this data, in fact, support the fact that PPm1K might be a really important actor in the pancreatic islet and maybe during type 2 diabetes. So we wanted first to evaluate if PPm1K was well expressed in the pancreatic islets. So we used different genetic database available online, and so compared the Tiger and GTEC database. And so thanks to the Tiger, which give us the opportunity to measure PPm1K in pancreatic islets compared to all pancreas, we saw that yes, PPm1K in the pancreatic islets is really highly expressed, as highly as, for example, the heart. More importantly, collaborating with Jeff Tessem in Provo, Utah, we were able to show that PPm1K mRNA expression are strongly downregulated in human type 2 diabetic islets compared to non-diabetic. And so this was really interesting, and once again confirmed that PPm1K might play an important role in type 2 diabetes, development and in pancreatic islets in particular. So as PPm1K gene was associated with insulin secretion genes, we really wanted to focus on the role of PPm1K in the beta cell within the islets. And using this single cell data from the Brisova lab, we were able to indeed detect high expression within the pancreatic islets, so in particular in alpha and beta cell. So we wanted then to know if PPm1K was absence of PPm1K could modulate beta cell function. So we use the insulinoma INS1 83213 pancreatic beta cell line, and we saw that after knockdown of PPm1K, we could observe as soon as 48 hours after transfection with siRNA, a decrease of EDU positive cells, so a decrease of cell proliferation, and even stronger at 72 hours. Associated to this, we also measured an increase of insulin secretion in response to glucose after PPm1K knockdown. So interestingly, this phenotype has been, similar phenotype has been explained before by an increase, in fact, of ER stress by high insulin production, and ER stress can lead to inhibition of proliferation. So we decided also to check in INS1 cells if maybe, in fact, PPm1K could modulate ER stress. And so we measured a subset of genes involved in ER stress, so UPR response. And indeed, after PPm1K knockdown in INS1 cells, we showed an increase of XBP1 spliced isoform and also HSPA5, so BIP expression. ER stress is a really important mechanism in the beta cell, but also ER stress can be deleterious and can unresolved ER stress can lead to beta cell failure, and also has been linked to beta cell loss of identity. And so screening different genes in different pathway, in fact, we realized that PPm1K was also leading to a decrease of MAFE expression and NK61, which are two markers of beta cell identity. And on the contrary, leads to increase of IRX and glucagon expression, which are markers for alpha cells. So at this point, we thought that, so yes, PPm1K modulates beta cell physiology, but it seems also that it's increasing ER stress and could be deleterious and in fact increase beta cell dedifferentiation. So then we really wanted to know by which mechanism PPm1K could induce this phenotype. So logically, we thought first that this should happen through modulation of branched-chain amino acid metabolism, so BCKDH activity. So we measured the phosphorylation of BCKDH E1 alpha, so which is a really good readout of BCKDH activity in INS1 cells. And unexpectedly, knockdown of PPm1K in INS1 cells didn't lead to the expected increase of E1 alpha phosphorylation. So after this, we decided to perform tracing experiments using KEV 13C KEV. And by doing so, once again, it was quite unexpected because we didn't see any enrichment of 13C in KEV or valine, but on the contrary, a slight but significant increase of 3-HIB. And 3-HIB is a product of KEV catabolism, so through BCKDH. So together, this data suggested, in fact, that PPm1K knockdown was inducing a slight increase of BCKDH activity. And this can be explained by a compensatory decrease of mRNA expression of BDK in INS1 cells. So BDK is the inhibitory kinase of BCKDH. So this data also suggested to us that PPm1K might not be the dominant regulator of BCKDH activity, BCKDH phosphorylation, and that maybe in insulin cells and probably in beta cell, PPm1K modulation is not going to induce a dramatic modulation of BCAA catabolism. This concept has been shown, in fact, several times in the literature. And for example, here, this study from Yibin Wang lab. And so PPm1K is described as a critical regulator of branched-chain amino acid catabolism. However, when you, for example, look in the liver of PPm1K knockout mice, so PP2CM, the gene coding for PPm1K, indeed, you see a really strong increase of phosphorylation of BCKDH in the liver. However, no differences, no effect is seen in the heart. So this really demonstrates that PPm1K might, in fact, depending on the tissue or the cell type, not be a dominant regulator of BCKDH. So in order to understand which other mechanism could be implicated, we went back to a phosphoproteomic study that we performed previously. And so this was done in the liver of rat overexpressing PPm1K. And what was really interesting is that, so the highest change of phosphorylation was not seen for BCKDH, but ribosomal protein S6 and on all known and really well studied phosphosites. And also what was really surprising is that mTOR and S6 kinase phosphorylation, so upstream of ribosomal S6, were not found in this list. So we started to believe that maybe PPm1K could be, in fact, a new and maybe one of the only non-phosphatase for ribosomal protein S6. So through its known role, PPm1K is already suspected to modulate mTOR activity. Indeed, PPm1K will dephosphorylate BCKDH, leading to a decrease of leucine availability, and this should decrease mTOR activity, decrease S6 kinase phosphorylation and activity, and decrease S6 phosphorylation and protein translation. However, we think that PPm1K probably is able to directly interact with S6 and dephosphorylate S6, inducing break on protein translation. So we use again our in vitro model, the INS1 cells, and indeed we could demonstrate in INS1 cells that knockdown of PPm1K does not induce an increase of phosphorylation of mTOR or S6 kinase in cells, but we could see an increase of ribosomal protein S6 phosphorylation and on all phosphocytes. Is this linked to a functional increase of protein translation? So we performed the sunset assay in INS1 cells. So this consists on incubating cells with puromycin. Puromycin is going to be incorporated in newly synthesized protein. And this can be done in presence of cyclohexamide to inhibit protein translation and measured by Western blots. And our data showed that knockdown of PPm1K increase incorporation of puromycin and so protein translation. And this was also quantified here. So this is really preliminary data, but we wanted also to perform ribosomal profiling. And these results suggest that PPm1K seems to increase overall ribosomal content. But really what is important is that PPm1K knockdown increase the polysome fraction. So polysome are the active ribosome, so active protein translation. Associated to this, we also could measure that knockdown of PPm1K induce an increase of protein content in cells. So at this stage, we wanted really to understand how PPm1K was regulating S6 phosphorylation. So first, we wanted to really validate that this was independent of BCKDH activity using a different approach. So using BT2. BT2 is a pharmacological inhibitor of BTK. Inhibition of BTK by BT2 should lead to activation of BCKDH, increase of utilization of branched amino acids, and so should lead to inhibition of mTOR. We also then wanted to check the ERK P90 risk pathway because P90 risk is another known kinase for ribosomal protein S6. So to validate that PPm1K knockdown was not inducing a phosphorylation of S6 through another pathway. Finally, we wanted, so in 3, 4, and 5, but by targeting different signaling pathway, lead to inhibition of mTOR. And this way, we were thinking that this would be really great to show that, in fact, PPm1K uncouple phosphorylation of S6 from mTOR S6 kinase signaling. So first, we used BT2 in insulin cells. So as expected, BT2 leads to a decrease of BCKDH phosphorylation, so activity, and this is similar in control in siRNA PPm1K cell. In the same extent, this also happened for S6 kinase. We can see a decrease of phosphorylation of S6 kinase for both control in siRNA PPm1K transfected cells. However, when BT2 decrease phosphorylation of S6, PPm1K knockdown still presents a higher phosphorylation of ribosomal S6 in insulin cells. So this demonstrated that effects of PPm1K on S6 are independent on BCKDH. Then we wanted to be sure that this was not due to activation of another pathway than mTOR. So we looked at the P90 risk pathway, so phosphorylation of ERK upstream of P90 and P90, and didn't really see any differential phosphorylation between control and siRNA PPm1K. So we concluded that PPm1K knockdown is not modulating ribosomal protein S6 phosphorylation through activation of this pathway. Then, as I told you earlier, we wanted to really show that these effects, PPm1K effects, were to uncouple S6 from mTOR S6 kinase. So by targeting different signaling pathway known to modulate mTOR activity. So first, we wanted to inhibit directly mTOR using rapamycin. We performed kinetic of rapamycin in insulin cells. Rapamycin leads to a rapid decrease and strong decrease of S6 kinase, both in control and siRNA transfected, PPm1K siRNA transfected insulin cells. Rapamycin also decrease ribosomal protein S6 in a time-dependent manner, And interestingly, PPm1K knockdown delays the effects of rapamycin. So then we decided to inhibit mTOR using an activator of AMPK, ICAR. So using ICAR, we could see that in fact, this was really interesting because cells were incubated for up to 12 hours. ICAR was decreasing S6 kinase phosphorylation, so showing that mTOR was inhibited, both in controlling siRNA PPm1K transfected cells. ICAR, so activation of AMPK, decreased phosphorylation of ribosomal protein S6 in insulin cells. But when PPm1K is knocked down, even after 12 hours of incubation with ICAR, we still observed a higher phosphorylation of S6. So this really was quite exciting and really showed that PPm1K, in fact, seemed to keep this high phosphorylation of S6 even in a context where mTOR is inhibited. So lastly, we also wanted to do the same by inhibiting the insulin signaling, so inhibiting PI3 kinase. So we used vortmannin. Voltmanine, as you can see again, so this lead to a decrease, is here as a control, decrease of AKT phosphorylation. This is happening both in controlling siRNA PPmK transfected cells, and we can also observe a decrease of S6 kinase phosphorylation. Voltmanine also decrease in a time-dependent manner S6 phosphorylation, and once again, PPmK knockdown delays the effect. So PPm1K really uncouple S6 phosphorylation from mTOR S6 kinase signaling. So it was really interesting, this experiment, because I noticed that PPm1K seems to decrease AKT phosphorylation in basal state here. And I was wondering if maybe PPm1K could alter insulin signaling. So to verify this, I treated insulin cells with IGF-1, and PPm1K not only decreased phosphorylation of AKT, so in untreated condition, but also reduce AKT phosphorylation in response to IGF. So then we wanted to know whether this was due to a defect of upstream signaling, a defect of IRS activation, defect of PDK1 phosphorylation, so one of the kinase phosphorylating AKT. So we looked at different phosphorylation of IRS, and in particular, which is really important, we looked at phosphorylation on serine 636, 639, which is a non-inhibitory phosphosite for IRS. And really no modulation no obvious modulation were seen between siRNA control and PPmK transfected cells. The same was also happening for PDK1 when we didn't see any differences, and so believe that this was not due to this activation of the pathway. So I started to think that maybe something else was happening in between, and in fact, so I was thinking that AKT maybe couldn't be recruited efficiently to the membrane. So you may know that when you activate the insulin signaling using IGF-1 or insulin, so this will lead to activation of PI3 kinase, and PI3 kinase will phosphorylate PIP2 to form PIP3, and PIP3 is essential to recruit AKT to be phosphorylated by PDK1 and mTORC2. But another actor, P10, phosphatase, can dephosphorylate PIP3, and increased activity of P10 will lead to decreased AKT signaling. So I decided to look at P10 expression, and in fact, we could see that P10 is increased, protein level of P10 is increased after knockdown of PPmK. And this can be also explained by, in fact, an increase of P10 mRNA expression. So then we treated cells with a vanadate compound inhibitor of P10 in order to show that the defect of AKT signaling was due to an increase of P10 expression and probably activity. And indeed, inhibition of P10 could rescue phosphorylation of AKT upon PPm1K knockdown. So finally, I wanted to share these few results with you. So we are really excited to describe and characterize what is the role of PPm1K in beta cell in vivo. So we are generating our model of beta cell specific PPm1K knockout mice. And so we are breeding our PPmK flox with inducible MIB-3 mice. And we did the first quick experiment. So this was in collaboration with John Campbell's lab. And so we treated mice with tamoxifen by gavage, and after 14 days, isolated islets and prepared the islets for cell sorting. And we validated that our model works because PPm1K is really only knocked down in the beta cell specifically. So here we can see a decrease of PPm1K mRNA expression in islets, not in alpha cells, but indeed in beta cell. We don't see any off-target effects in other tissue. And what is interesting, so even if this is really at early time points after knocking out PPm1K, we don't measure any difference of circulating branched-chain amino acids, so confirm what we believe is happening in Insulin cells, no modulation of branched-chain amino acid catabolism in beta cell. And more interestingly, already at these early time points, we see that knockdown of PPm1K induce an increase of the ratio XBP1 spliced to unspliced in pancreatic islets in mice. So we think that maybe the mechanism seen in Insulin cells is already taking place in vivo. So as a conclusion, our working model, we showed that PPm1K is a really important regulator of beta cell proteostasis. So by regulating ribosomal protein S6 phosphorylation, PPm1K acts as a brake on protein translation. And PPm1K might be an important regulator of beta cell health. However, when PPm1K expression is decreased, so as we showed, it's happening in pancreatic islets in human during type 2 diabetes. This might lead to hyperphosphorylation of ribosomal protein S6, increase of protein translation, and modulation of insulin secretion. As we showed in INS1, increase of insulin secretion. Altogether, this increase ER stress in beta cell, and this play probably an important role during beta cell failure. We don't really understand how this other pathway connects, but we think that increased protein translation could increase PTEN, leading to decrease of AKT signaling. And what is really interesting is, in fact, we can connect this decrease of AKT signaling that we observed in INS1 after knockdown of PPm1K to also the possible loss of beta cell identity that we showed with a decrease of NK61 and MAFE expression and increase of IRX and glucagon expression. So I would like to acknowledge all these people on this line. So the White Magara lab, and so in particular my PIs, Philip White and Rob Magara. Also, so other people in the Duke Molecular Physiology Institute, so collaborating, working with us. And Jeff Tessem in Provo in Utah, at BYU, and his team who are collaborating with us. And if you want to reach out and ask any question about this presentation, you can do this on Twitter, and thank you.